Hi, my name is Patricia Valdespino. I am a scientist at the Berkeley Synchrotron Infrared Structural Biology Imaging Program at the Lawrence Berkeley National Lab. I will talk about our studies on the Antarctic microbes that thrive during the polar night, which is the darkest and coldest period of the Antarctic calendar. Some areas of Maritime and Peninsular Antarctica are exhibiting the highest temperature increase in the southern hemisphere. As an example, at Esperanza Base on the Antarctic Peninsula, the temperature rose to a record breaking high of 18.3 Celsius degrees in 2020. Antarctic research is helping to monitor, prevent, and mitigate the effects of climate change and global change. Temperature rise impacts ecological and biogeochemical functions as well as biodiversity. Microbes are the most diverse component of Antarctic ecosystems. They play key roles in ecosystems' function, adaptation, and resilience. Microbes control flux of matter and energy in these low temperature, low nutrient ecosystems. The microorganisms we study develop in the summer and cannot migrate during the winter. They remain in inland Antarctica under ice and snow during the polar night, which typically lasts more than 6 months. Because of unpredictable and severe weather, Antarctic research has focused on the summer. Here you see a sampling effort of a summer campaign. But we have limited knowledge of processes occurring during the polar night. For example, we would like to know more about the active diversity and the fluxes of matter and energy in the ice and snow caps. In this study, we integrated metabarcoding, metagenomics, and synchrotron infrared imaging to investigate inland microbial mats, which typically develop in streams and ponds during the summer. According to our results and previous work, the low availability of nutrients such as carbon, nitrogen, and phosphorus frequently limits life. Our results show that spring mats, the only ice-free samples, are dominated by photosynthetic phototrophs, mainly cyanobacteria. In contrast, during the polar night, mats are dominated by heterotrophs of four bacterial groups. The overall diversity of Antarctic mats is remarkably high, evidenced by the large number of taxa comprising less than 1% of the DNA community composition. Activity during the polar night, here shown in a dark box, is limited to a few microbial taxa, such as some proteobacteria, firmicutes, and bacteroidetes. Metabolic potential differences are particularly marked between autotrophs and heterotrophs, regarding both cellular structure and function pathways. Heterotrophs depict a large and diverse genetic potential, for example, for amino acid recycling. Infrared imaging shows structural and chemical differences between well developed and early stage ice covered microbial mats. A common chemical feature found was, for example, the presence of aromatic compounds in the studied biofilms. By understanding how Antarctic microbes respond and adapt to the environment, we aim to help predict and mitigate the effects of global change. This research is a cooperative effort among several research groups in the US, Mexico, and Argentina. Thank you.